Hi everyone, and welcome to your first screencast of biology. We will be using these videos to help learn new material and also so that you can go back and review things in case you missed out on the first time. So without any further ado, let's jump right into the scientific method. I know most of you have studied the scientific method for many, many years. It certainly isn't new information. So we're going to go through this pretty quick. In general, when I talk about the scientific method, all I really mean is a systematic way of solving problems. We use the scientific method to create a series of steps to solve a problem or to answer a question, and then have that information available to other scientists. We always start out with some kind of observation, and an observation is a natural occurrence or something that you see that has no explanation. You don't know why it's happening. And we're going to use the example that plants will grow under a light bulb. So, you see plants all the time, even some in our classroom, that if I turned all the lights off and just put a light over one of them, it would still continue to grow. And we've always learned that plants need sunlight to grow. So, what's going on there? I did want to note this is one of my favorite quotes. The most exciting phrase to hear in science, the one that heralds new discoveries, is not Eureka, but that's funny, by Isaac Asimov. And it's a wonderful quote because it's completely true. When we see new discoveries in science, it's very rarely a breakthrough discovery that everyone says, oh my gosh, we just discovered this amazing thing, and everybody jumps for joy. Usually what happens when big breakthroughs occur is a scientist or a team of scientists sees a set of data that they don't expect. And they think, hey, that's kind of weird, and they start to investigate. And that's when we come up with some of our best discoveries. Once I see this odd occurrence, once I observe something, the very next thing I'm going to do is research. And this is a step that a lot of people leave out or they forget about. I can't go from watching my plant grow under a light bulb to just immediately setting up an experiment. That is a very simple experiment, but when we look at more complex things, you really have to do your background research. So you're going to learn as much as you can about your observation from other scientists. You go into the published literature and you start to do a lot of research, a lot of reading. An example for this, we would come up with some research and find out that plants use photosynthesis to make sugar and grow, and the chemicals necessary for photosynthesis take in light and convert that into chemical energy. So after I've done all my reading, I realize that, okay, plants do photosynthesis and plants need light. But it doesn't specify what kind of light. So now I'm curious and I have a question, and that is what's going to start off my experiment. My problem, and I don't have a separate slide for this, but my problem for this would be what kind of light do plants require to grow? Once I have a question, I need to come up with a hypothesis. I know many of you have learned the term for the explanation for a hypothesis is an educated guess, and that's kind of true, but I want to be a little more specific. A hypothesis is a testable explanation for your observation, and it has to be based on your research. You can't just throw a idea out there and call it a hypothesis. A lot of times it does use the if-then structure. But not always, and we'll see some examples where that doesn't happen, but that is generally the more simple experiments. So for our example, if a plant is placed under a red light, then it will not grow as much as a plant under a white light. That would be an acceptable hypothesis. Notice that it is testable. It is something that I can do an experiment to prove or to test. I do want to pause here and talk about variables. One of the most important things, and often the most difficult things for students, is to know the difference between an independent and a dependent variable. An independent variable, starting with an I, are those that I, as the scientist, have control over. I can change those. We always know them at the beginning of an experiment, and usually that is what we are testing. So for our example, the independent variable is the light color. I can change it. I have decided to put a red light bulb in my lamp or a white light bulb in my lamp. and I already know those colors. It's not up in the air yet. So that is my independent variable. My dependent variable literally depends on the experiment. They're not known until the end of the experiment, and that is what we are trying to measure. So in our experiment, the dependent variable is going to be the plant growth. I don't know yet. That's what I'm trying to figure out. So instead of just saying plant growth, I would like us to be a little bit more specific and state how we're going to measure it. So in our case, let's go with plant height. We also have to have a control, and a control can be called many things. We call it a control for short. A control group is the proper term. We also call it the norm. Basically, it's one of your experimental setups where we don't change anything. Nothing is changed beyond what would normally happen. And we have to have a control group because we need it for comparison. In the example I have here, if we only measured plant height under a blue light bulb, an orange, and a red, we would get data, but I'd have no idea how tall the plant should normally grow. So we have to include a control group to compare our colored light bulbs to what would be considered normal. So our control group in this experiment is going to be the white light, which is normal light. All right, so now that I've got all my variables set up and my control, I'm going to run an experiment. And an experiment is nothing more than an organized test of your hypothesis. You must always have an independent and dependent variable and a control for it to be a legitimate experiment. Here's an example. Notice my experiment has a procedure, and the procedure in this case has been split into two sets. I have my materials and my steps. So let's look at them. We have 3 pea plant seeds. Sorry, that's a typo. Those are plants, not pants. 3 pots, 600 milliliters of water, 3 lamps, a red bulb, a blue bulb, a white bulb, and 300 grams of soil. Notice that each plant gets the same amount of soil, the same amount of water. Those are controlled variables. It's not a control group like we saw before, but they are variables that we are now controlling or keeping the same so that I know the results that I see will be from only the color and light, only the light difference. When I get down to my steps, I have that I'm going to fill each pot with 100 grams of soil, put a seed in the middle of each pot, and place each pot under a different colored bulb. Water each plant with 100 milliliters of water each day for 20 days. Every 7th day, record the height of the plants. Notice that my directions are very clear. They're step by step, and they include specific measurements for everything we do. It's very important when we start writing our experiments that you list your materials and you list your procedure in a way that anyone else can follow. I should be able to take your procedure and figure out exactly how much of any material you used and figure out exactly what you did to get your results. Remember, science, good science, has to be repeatable, which means we have to be able to communicate what we're doing effectively, and we'll get more into that later. All right, so after my data collection period, after my actual experiment, I'm going to go into analysis. This is also the results, and when we do a lab report, you may end up calling it the results section. Here you're going to collect your data and organize it. You can't just throw a bunch of numbers on a paper and call it data. You have to put it in a way that someone else can understand. A lot of people are going to use charts or graphs or some sort of pictorial representation to present their data. But you also include your raw data, your numbers themselves. This is an example of the data collected from that experiment. You can see in week 1, week 2, and week 3 the different plant heights that were recorded. In this case, it would be expected for us to graph this data to make it a little bit easier to understand or to see it in a snapshot. And when we graph this, and we will do some graphing practice in class, in general, the independent variable is plotted on the x-axis, and the dependent variable is usually on the y-axis. That's not a hard and fast rule, it's just a generality that you normally see. So in this case, remember our independent variable was the light color. So I'm going to put my light colors, the white, the blue, and the red, on the x-axis, and my centimeters, my height, on the y-axis. We'll also talk about why we use different graphs in different situations. Now that you have your data, you can't just leave the data alone. We have to come up with some kind of conclusion. So based on our experiment, you have to summarize your findings. This is actually a very short section. Believe it or not, the analysis goes into a lot more detail. The conclusion, I don't want you to just restate your hypothesis or accept or reject your hypothesis. Tell me what you found. And the example I have is actually quite short, but there's also a lot of room to talk about new ideas or new, maybe you got something you didn't expect. And in your conclusion is where you're going to present other scientists with the idea that, hey, maybe we should do some research into this area, or maybe we should repeat the experiment and see what happens again. So the conclusion does a lot of different things, but in general, it's going to wrap everything up. The example I have here, the data shows that overall the plant in the white light grew taller than the plants in red and blue light. The plant in red light grew 1 centimeter taller than the plant in blue light and showed earlier growth. Therefore, light color does affect plant growth. I accept my hypothesis. Again, they've summarized everything nicely for me. They told me what they found. If this student wanted to go a little further, maybe they want to know why red light is more effective than blue light, and maybe that's an area for further study. And finally, this is a step everyone generally forgets. Scientists have to publish. We never do science in isolation. And what I mean by that, we always think of a scientist as this guy in a white lab coat working away by himself, and never hoarding all of his results and then coming up with these big discoveries. What really happens in the scientific world is scientists work together all the time, and they cannot sit on their results. Instead, they publish, they write papers. So when they discover something or come to any kind of conclusion that is scientifically important, they need to share those findings with the scientific community. And we do this through a process called peer review. They submit their papers, and other scientists read them, review them. Other scientists may choose to copy their experiment to validate it. Because the more times we see the same results, the more valid an experiment becomes, or the more valid a hypothesis becomes. They may decide that, hey, something was wrong in your methodology, something, you didn't account for something, and this is how we refine science. And they also build upon each other. So isolation is bad for science. We have to work on communication and on proper writing skills. Scientists are very, very much writers. From personal experience, I was a science major in college, and I minored in English and spent a lot of time in both departments. And I can say without any hesitation that I wrote more in my science courses through research, through publishing findings, and communicating with with my peers at the university. You end up spending a lot more time doing research and writing as a scientist than a lot of people realize. So that's something we're going to be looking at, and we're going to be building that skill throughout the semester. So make sure I think that's the end, yes. Make sure you go back through this. If you would like to print off these notes and make your own little notes in the margin, you may. If you like to listen and write down as you go, that's perfectly fine as well. But please do make sure that you have this information in your notes, because you will need it for class next time. Thank you.